Welcome to Bio 100 Lab. The purpose of this lab course is to provide hands-on experience with biology concepts in a laboratory setting. This course will enhance your understanding of topics covered in your lecture course and will also present new concepts. Biology is an experimental science that continues to evolve as scientists test their ideas. This lab course should give you a sense of how biologists use observation and experiments to learn about the natural world. Your lab will be taught by either a professional laboratory instructor or graduate teaching assistant. Please make note of his or her contact information. Your instructor or TA will hold regular office hours in order to help you outside of class, in addition to answering questions during class. In today's lecture, we will take a look at the course overview and what to expect for the semester, review the syllabus, including grading structure, examine and understand the lab safety rules, define and learn to prevent academic dishonesty, and we will delve into the first lab, the scientific method, where we will define science and the scientific method, how to control an experiment, and briefly go over the lab exercises. Course overview. Please purchase the required laboratory manual for this course at the NAU bookstore. You won't need a lab coat, goggles, laboratory notebook, or any other special equipment. Your syllabus is your guide to the course. Please read it carefully. It explains the assignments and tests and provides the schedule for labs. Labs will not be done in the same order they appear in the laboratory manual, and they will not follow the structure of your lecture course. You are responsible for reading and understanding the entire syllabus. You can earn your first points in this course by filling out, signing, and returning the syllabus contract provided by your instructor. This contract represents your promise that you have read and understood the syllabus. Please ask your instructor if you have any questions about the syllabus. Safety rules are on the inside cover of your lab manual. Consider these your code of conduct for lab. Break any of these rules and you will lose professional points and may be asked to leave the lab. Semester expectations. Attendance is mandatory. Please be on time. Quizzes or exams will take place at the beginning of the class period. If you are late, you may miss your opportunity to take the quiz or test. No makeup labs will be given. If you must be absent, please notify your instructor before lab. You are responsible for coming to lab prepared to do the activity, meaning you must read the lab manual chapter for the week's lab prior to class. There will be at least one question on the weekly quiz covering material in the lab that you are about to do. You and others are safer in the lab if you are prepared to do the activity and are not fumbling around because you have not read the activity carefully. You are responsible for completing all assignments on time, following all safety guidelines, cleaning up after activities, and asking questions if you are unsure of anything. You must be cooperative with others and behave in a respectful manner. If you are disrespectful or uncooperative, you will be required to leave and may be dropped from the class. So, what does a typical lab look like? We'll start with the agenda. The day's agenda will always be written on the left side of the board. It will let you know what we will be doing that day and the order in which we'll be doing all lab activities. The agenda may also include reminders and due dates as necessary. Next is the quiz. The quiz will take approximately 10 minutes and will occur at the beginning of lab. If you're late to lab and the quiz is still out, you will not receive extra time. If you're late to lab and the quiz is over, you will not be able to take the quiz. So be sure to be on time. The majority of quiz questions will be about the lab completed the previous week, with a few questions about the lab to be completed that day. Lecture. Read the entire lab exercise to be completed that day before coming to class. Remember, you're going to be quizzed on that material. If you have a question during the lecture, feel free to ask it. If you are unsure how a lab activity is to be performed, ask your question at the appropriate time during the lecture. Next are your lab activities. Lab activities may be done out of order, so pay attention to the agenda and to the TA during lecture. Answer questions in your lab report as you go. Next is the cleanup. You are responsible for the cleanliness of your lab table. Essentially, it should look exactly the same as it did as when you arrived. If your area is not clean when the TA comes over to review your lab report for checkout, you will be asked to clean before checkout can occur. You will lose professional points if you do not clean up after yourself. Remember, there are a lot of students that use this area, and it's essential to help keep it clean. Lastly, there's checkout. Once your table, or your side of the table, has answered all questions in the lab report and have cleaned the lab table, call your TA over. The TA or instructor will review the lab report, checking your answers for accuracy, clarity, spelling, and basic grammar. Yes, this means you will need to write in complete sentences whenever possible. If your bench is clean and your answers are acceptable, the TA will initial, sign, or stamp your lab report. This stamp ensures that you will receive credit for the lab report. Unfortunately, academic dishonesty, or cheating, is a topic we must discuss. Any cheating can result in an F grade, and cheaters may be expelled from the university. Cheating is not only wrong, it's just not worth it. Cheating is defined as any attempt to gain an unfair advantage over one's fellow students. Types of cheating include plagiarism, fabrication, and fraud. Plagiarism is any attempt to pass off another's work as your own, such as copying a term paper or parts of a term paper written by someone else. Fabrication is any attempt to present information that is not true. Fraud is any attempt to deceive an instructor or administrative officer of the university. Unlike fabrication and fraud, students sometimes plagiarize without realizing it because they have not been taught how to recognize and avoid plagiarism. A university is an institute of higher learning where the highest standards of academic integrity are upheld. Do not rely on a definition of plagiarism that may have been acceptable in high school, but is not the accepted definition for university. Sometimes plagiarism is deliberate, and sometimes it's accidental. Understanding more about what plagiarism is will help you prevent accidental plagiarism. The following tips on how to write an honest paper are from Charles Lipson's excellent book, Doing Honest Work in College. 1. Cite others' work whenever you rely on it. 2. When you use someone's words, quote them accurately, mark them as a quotation, and include a citation. 3. When you paraphrase, use your own distinctive voice, not that of the author's. Be sure to include a citation. 4. Never represent anyone else's work as your own. 5. Never hand in the same paper to two classes unless you have permission from both instructors. 6. Never buy, sell, or borrow papers. Do your own work. NAU's Klein Library offers resources for students who are writing papers. Ask the librarians or look at the Klein Library website. Now, let's take a look at lab rules and safety. Look at the inside cover of your lab notebook for the listed laboratory rules and safety. Number 1. Locate the first aid kit, eye wash station, fire alarm, building exit, and telephone prior to the beginning of the first lab activity. Be familiar with how to use the equipment in the event of an emergency. Number 2. No eating or drinking in the laboratory. Number 3. Familiarize yourself completely with the details of each experiment before coming to each laboratory. This is done by carefully reading the appropriate chapter in the lab manual prior to lab. Number 4. Microscopes are precise and delicate instruments that need to be handled with care. Carry and use microscopes according to the methods demonstrated in each class and report any problems with microscopes to the instructor. Number 5. If something breaks or spills, notify the instructor. Do not attempt to clean it up yourself. Number 6, in case of injury, notify the instructor immediately. Number 7, put waste into the appropriate container according to type: broken glass, dissection material, chemicals, or non-contaminated waste and paper. Ask your TA where these different containers are. 8, wash your hands with soap and water upon arriving and before leaving the laboratory. Remember, this equipment is shared by a lot of students and it's important to keep it clean. 9, wear closed-toed shoes and tie back long hair. Your instructor may ask you to leave if you come to class in sandals. 10, coats and other materials are not to be placed on the lab tables. Place personal items in the drawers of the lab tables. In lab, you will be moving around a lot, and it's essential that your personal items are out of the way. 11, return all materials to their appropriate storage area and clean your laboratory station prior to leaving class for the day. Be sure to ask your TA if you have any questions or concerns regarding the laboratory rules and safety. Next, we will go over the syllabus. Labs will not be completed in the order in which they appear in the book. Thus, it is important for you to know which lab we will be completing each week. This information, the schedule, can be found in the syllabus. Due dates are also listed in the syllabus. Due dates are very firm, so even if the university is closed, your work is due. You can send it by email or whichever way your TA prefers. Now let's take a look at the grading structure for the semester. Attendance is mandatory. You will earn up to 5 points for completing the questions in your lab manual for each lab. We will also have weekly tests. They're worth 10 points each and cover last week's material and a couple of questions from the upcoming lab. A good way to study for these tests are to review the PowerPoints on BB Learn. There is one large assignment for the semester, and it's the research article hypothesis and critique. It's worth 50 points. We will talk about this more next week. There's also one opportunity for bonus points throughout the semester from the current science news presentation. Once again, we will discuss this further as it comes up. The best way to have academic success this semester is to be prepared for each class. Read the manual ahead of time and review the PowerPoint. Also attend every lab and actively participate. Don't let your lab partner do all the work. The purpose of lab is to have a hands-on experience, so take it. Be sure to study for the weekly quizzes or tests. Also, put in full effort for the research article assignment. It's worth 50 points and is very important. Email is a very important method of communication. Be sure to regularly check your NAU email for announcements and assignment information. Please contact your instructor if you have any questions or concerns with the class or need help with any material. Okay, now that we've covered the structure, grading, and syllabus and expectations of the class, we can delve into the first lab, the scientific method and the nature of science. The objectives for each lab can be found at the beginning of the chapter in your lab manual. By the end of this laboratory, you should be able to name the steps of the scientific method, distinguish between qualitative and quantitative observations, define data and hypothesis, distinguish between an observation and an inference, be able to state an experimental hypothesis based on observations and state the null hypothesis, and construct an experiment using the scientific method. Be sure to check in with your objectives upon completion of the lab. So, what is science? Science is a process of obtaining new knowledge about the natural world using well-defined, systematic, repeatable methods. The scientific method is the standard, well-defined, systematic method and has been used by generations of scientists. A valid scientific result is one that is reproducible. A different scientist who follows the same series of well-defined steps of a particular experiment as the original scientist should obtain the same or nearly the same result. If the result cannot be reliably reproduced, it is not accepted as valid. This is one of the great strengths of the scientific approach. So, what is biology? Biology is the study of life. You are alive. So, as you study biology this semester, remember that you are biology too. In the words of John Steinbeck, the true biologist deals with life, with teeming, boisterous life, and learns something from it, learns that the first rule of life is living. Now we'll move on to the scientific method. The modern scientific method grew out of the thought and work of many philosophers and scientists from Aristotle to Roger Bacon to the present day. The scientific method consists of 5 steps that are often repeated in a cycle as more information is learned by the experiments. The 5 steps are observation, question, hypothesis, testing or experimenting, and conclusion. The first step of the scientific method is observation. But what is an observation? An observation is something that you view, see, or otherwise experience. Conversely, an inference is an assumption you make based on the observation. For example, you may observe that the jaguar in this picture has tan fur with dark spots. You may infer that this is to help the jaguar camouflage and aid in hunting. There are 2 basic types of observations, qualitative and quantitative. A qualitative observation is a descriptive observation where no actual measurements are made. These observations usually lead scientists to look into something more quantitatively. For example, the bird in this picture is yellow and black. A quantitative observation is an observation that involves some form of measurement. This type of observation is more precise than a qualitative observation and is used more often by scientists. For example, the bird has a 40-centimeter wingspan. The bird has a beak that is 2 centimeters long. The bird weighs 7 grams. Quantitative observations are easier to analyze, but qualitative observations are also important. For instance, if you are a scientist studying birds, you may notice that a bird of a certain species has different color eyes than any other bird of that species. Is this a mutation, you may wonder? This could lead you to more studies. These observations then lead to questions. For example, why can birds of the same species have different colors? Why are some birds more colorful than others? Does temperature affect the rate of butterfly development? Can you think of another question that comes from an observation? Maybe something along the lines of, how quickly does your heart rate return to normal after it exercises? Is it faster for people who exercise more? After observing and questioning, the next step in the scientific method is to formulate a hypothesis. A hypothesis is an educated guess or tentative explanation for an observation. For example, you are heating up water to make tea, but you're in a hurry. You hypothesize that increasing the heat setting on the stove will decrease the time it takes for water to boil. This is your experimental hypothesis. Every hypothesis has a twin that is its opposite. This is called the null hypothesis and abbreviates as HO, while the original hypothesis is called the experimental or sometimes the alternate hypothesis, abbreviated HA. So, our null hypothesis that pairs with this experimental hypothesis is that increasing the heat setting on the stove will not change the time it takes for water to boil. Remember, the null hypothesis means no change. Let's try another example. A chicken farmer may have recently introduced a new brand of food and noticed that suddenly the chickens are laying more eggs. He might hypothesize that the new food is causing chickens to lay more eggs. In the chicken feed example, the experimental or alternate hypothesis is that chickens eating the new feed will lay more eggs than those eating the old feed. Its opposite, the null hypothesis, is chickens eating the new feed will not lay more eggs than those eating the old feed. Notice that every possible outcome is covered by our alternative and null hypotheses. Having clearly stated the experimental and null hypotheses helps when designing an experiment. You must design it so that the outcome will support either the experimental hypothesis or the null hypothesis. If it doesn't, it is not a well-designed experiment. With a well-designed experiment and clearly stated hypotheses, it is easier to determine what the results mean. When you develop a hypothesis, make it sound like a statement of fact. Chickens eating the new feed will lay more eggs than chickens eating the old feed. To write the null hypothesis, simply add the word not or words not change to the sentence. Chickens eating the new feed will not change the number of eggs laid. Or, chickens eating new feed will not lay more eggs than chickens eating old feed. Both are acceptable. Next, you'll want to test your hypothesis in a controlled conditions. This is the experiment. A good experiment tests the hypothesis. This may seem obvious, but if you do not have a clear hypothesis at the outset, it is hard to design a simple and elegant experiment that tests it. Good experiments only change one factor. If more than one factor is changed in an experiment, you cannot tell which factor caused the results. Good experiments include a control. This is a group of experimental subjects, such as chickens, in which nothing has been changed. It serves as a baseline measurement to which you compare the result of your experimental treatment. For instance, using the chicken feed example, the farmer hypothesizes that chickens fed the new feed will lay more eggs. But more eggs than what? More eggs than chickens that are fed the old feed. The chickens that are fed the old feed are the control, while those that are fed the new feed are the experimental group. A controlled experiment also eliminates as many differences between treatments and trials as possible. What if the farmer played soft music for the chickens that are fed the new feed and does not play music for the chickens eating the old feed? Because the farmer has changed more than one factor in his experiment, he cannot know if his results are due to the new feed or the soft music or both. It is vital to the experiment to control for all factors, even if you doubt their effect on the results. A recent study by McGill University concluded that male researchers induce more stress on mice than female colleagues, a factor that is not often controlled for in experiments. So, in your experiments, take the extra step to ensure a controlled environment. Even the slightest differences may skew your data. When an experiment is completed and the data, which is the information provided by the experiment, in our chicken feed example, the data would consist of the number of eggs laid by each group of chickens. When that data has been collected, the scientist uses it to determine which hypothesis, the experimental or the null hypothesis, is supported by the data. This is making a conclusion about the experiment. Using the chicken feed example, suppose the control group chickens laid an average of 2 eggs in 3 days, while the chickens receiving the new feed laid 1 egg in 3 days. The data, 2 eggs versus 1 egg, allow the farmer to conclude that chickens eating the new feed do not lay more eggs, supporting the null hypothesis. Suppose the control group chickens laid an average of 2 eggs in 3 days, while the chickens receiving the new feed laid 5 eggs in 3 days. This data supports the experimental hypothesis. But suppose that both sets of chickens laid exactly 2 eggs in 3 days. Which hypothesis is supported? The correct answer is the null hypothesis. Let's look at the movie Supersize Me to further examine the scientific method and experimental control. In the documentary, the director, Morgan Spurlock, eats McDonald's to examine the health effects of a fast food diet. His experiment in the movie started with an observation. People in the United States were frequently and severely overweight and ate a lot of fast food. Next came his question. Was fast food the major cause of the rise in obesity? His experimental hypothesis was that eating fast food will cause a person to gain more weight than a home-cooked diet. The null hypothesis would be that eating fast food does not change the amount of weight a person will gain. The testing comes next. Morgan ate McDonald's 3 times a day for 30 days and had to say yes to each and every offer to supersize his meal. After 30 days, he gained about 25 pounds and his cholesterol level rose dramatically to 230. Morgan then concluded that a fast food diet is detrimental to health and causes weight gain and increased cholesterol levels. However, there is a major issue with his experiment. There is no experimental control, no control group, no set amount of exercise or caloric intake, and no replication. It is just one man eating a variety of McDonald's meals for 30 days. While it was an interesting social experiment, it is not a valid scientific experiment. So, what would be a better experiment? First, you need both a control group, getting no treatment or change, and a test group. A larger study group is also desired since there may be individual genetic differences. Having 2 groups of 15 lab mice is a good start. The control group gets fed regular mice food, and the test group eats a diet of McDonald's. Caloric intake and exercise must be kept controlled as well. Offering food and exercise to both groups for the same amount of time each day is a good approach. Only after you have a well-controlled experiment can you be confident in your data. Monitor the weight gain, body fat percentage, and cholesterol levels of the mice for an extended period of time. Let's stick with 30 days. Lastly, it is very valuable to compare your findings to other studies. Is your data an anomaly, or have other similar research projects come up with the same results? It cannot be stressed enough. Only a controlled experiment is valid in the scientific community. And the only way to have a controlled experiment is to have one variable changing while all others remain constant. Okay, now that you have all of the background information that you need, let's get started on the first lab. In exercise A, you'll have a list of statements and you'll determine whether those observations are quantitative or qualitative. In exercise B, you'll be looking at a box with 3 and only 3 objects. You'll determine what is inside the box without opening it. You have a list of potential objects and the instruments available to you. For exercise C, you will be conducting an experiment using the scientific method. Be sure to eliminate sources of error to create the most controlled environment possible. Potential factors to consider are left hand versus right hand, age of the penny, and size of the penny. Think about your sample size. Is testing 1 penny enough to make an inference about all pennies? How many pennies should you test before making an inference about all pennies? Why might pennies differ in water holding capacity? And one last thing. When you're completing your experiment, be sure to record your data in a legible table. Look at the table provided as a template.